Welcome back, students of 4th SEM, to your second class on one gene, one polypeptide, which is included in your CC10 syllabus. In the early 1940s, Biddle and Tatum developed the concept of one gene, one enzyme, which was later on modified in several ways. As I have told you in the earlier class that genes form mRNA molecule which codes for protein. Now sometimes several genes form a single mRNA molecule and then it is called polycistronic. Now from a polycistronic mRNA several polypeptides may be formed and these polypeptides form proteins. Some of the proteins may function as enzymes. So all proteins are not enzymes, but all enzymes are proteinaceous in nature. Now later on it was found that many enzymes or structural proteins are made up of subunits. That is, they are hetero multimeral. They contain two or more different polypeptide chains, and each of these polypeptide chain is encoded by a separate gene. As for example, in E. coli, an enzyme tryptophan is a heterotetramer. Why? Because it is composed of two alpha polypeptides, which is encoded by tryptophan A gene, and two beta polypeptides, which is encoded by tryptophan B gene. Similarly, in humans, the hemoglobin which transports oxygen from the lungs to the different tissues is also composed of two alpha globin polypeptides and two beta globin polypeptides. The alpha globin polypeptides are encoded by HbA alpha gene, which is located on chromosome 16, and it has 141 amino acids. Two beta globin polypeptides encoded by HbA beta gene located on chromosome 11, it contains 146 amino acids. Now if you look into this picture of hemoglobin, you will see two beta globin polypeptide chains, two alpha globin polypeptide chains as I have told you, and four non-proteinaceous iron containing heme groups. So from this it became clear that one gene, one enzyme concept is not perfect. It was modified to one gene, one polypeptide concept. This is more well illustrated in sickle cell anemia, which is prevalent among the Negroes. They have a mutant hemoglobin. Now look into this picture of RBC or erythrocytes. This is a normal RBC from a normal individual having a biconcave disc-shaped structure, whereas this one is from a sickle cell anemia patient which has the RBC of sickle shaped, elongated and curved. In afflicted individuals, the RBC aggregates in the capillaries of the veins at low oxygen tension and sickle cell anemia develops. As a result, several tissues may be deprived of oxygen and patients suffer from severe damage. At this stage, the sickle cell individual experiences sickle cell crisis, which may be fatal if untreated, because the RBCs are destroyed rapidly. The molecular reason lies in the difference of arrangement of amino acid molecules in hemoglobin. The sickle cell hemoglobin differs from the normal hemoglobin in the amino acid content. The sickle cell hemoglobin contains valine at the 6th position, whereas the normal hemoglobin contains glutamic acid at the 6th position. Now, this disease is inherited as a Mendelian trait. There are 3 types of genotypes controlled by a single pair of allele, that is HBA and HBS. The normal individuals are HBA, and affected individuals are HBS, having homozygous genotypes. Now this diagram shows the hemoglobin derived from HBA, that is a normal individual, and HBSS, that is sickle cell individuals, after electrophoresis of the blood sample was done, and then fingerprinting was done, and then amino acid analysis was carried out. Now, as you can see, when electrophoresis is done, the migration in case of normal individual towards the anode is more, that is negative, indicating that the negative charge is more, whereas in case of sickle cell anemia, the migration is less compared to that of the normal individual. And AS, a heterozygote showing a normal phenotype, here you can see both the types of migration. Now, when chromatographic study was carried out, you can see the altered peptide in the sickle cell anemia patient. Now, when the molecular analysis was carried out in between the normal individual and the sickle cell anemia, it was shown that at the 6th position in normal individuals there is glutamic acid, whereas in sickle cell anemia patient there is valine, that is glutamic acid is replaced by valine. Now, this is due to a single change or mutation in one gene. In the normal individuals, AT is present in DNA, whereas in the sickle cell patient, this AT has been replaced by TA. Now, from this DNA, when mRNA is being formed, then this is being from TA is formed, that is GAG, and from A, U is formed, so GUG. So, when polypeptide is formed from this mRNA, that GAG codes for glutamic acid. G stands for guanine, A for adenine, and G again for guanine. But in this case, that is in a sickle cell patient, you have GUG, which codes for valine. And this occurs at the 6th position. So normal individuals which have HbA are homozygous genotype and sickle cell anemia which has HbS, they are also homozygous, having a different amino acid at the 6th position. So we can see or we can conclude that a single change in one of the two polypeptide chain occurs due to a single mutation in one gene, as I have shown you in the previous slide. So from this we can conclude that one gene controls the synthesis of one polypeptide chain and not the complete protein. So this shows that one gene, one polypeptide concept is correct instead of one gene, one enzyme.
I hope students you all have understood one gene, one polypeptide concept. If you all have any queries, you all can always place the questions through the college portal and I'll be very glad to answer your queries. Thank you very much.